Delaware Tech, Chemistry 100, Chemistry 110, buffers. So buffers are solutions that resist changes in pH. In other words, when you add small amounts of a strong acid or a strong base, the buffer will prevent large changes from pH from occurring. And when we talk about strong acid, we mean an acid that when you add it to water, all of the acid will be turned into protons that will be released and will react with water to form hydronium ions. And typically when you see this written in most videos and most textbooks, they'll just show you the acid releasing H plus, a proton, but the reality is H plus is never by itself. It's always added to the water to form H3O plus. So anytime you see H plus, think H3O plus, because the acid's reacting with the water. And likewise, if you add a strong base to water, that base will interact and all of that base will be turned into OH minus, hydroxide. So strong acids will generate the same amount of protons or hydronium ions, and strong bases will generate the same amount of hydroxide, depending on how much acid or how much base you add to the water. And buffers, the role of buffers is they react with these two ions, the acid ions or the base ions, and prevent them from changing the pH of the water. So buffers come in two major categories, but they really are made of the same two components, a weak acid and a weak base. So what do I mean by weak acid? An acid that doesn't completely lose all its protons when it's put in water. And a weak base is a base that doesn't abstract all the protons for every base that's there. In other words, weak acids, only a small fraction of them become their conjugate base, and weak bases, only a small fraction become their conjugate acids. So we mix the two together, a weak acid and a weak base, usually by taking the weak acid and its conjugate base, or taking the weak base and adding its conjugate acid. And buffers are typically named for the base component. So they're always going to have two components, a weak acid and a weak base, and we usually name them after the weak base. And we'll go through examples of that later. So one way to make the buffer is start with a weak acid and add its conjugate base, and that will be a weak base as well. And so an example here in red is acetic acid, CH3CO2H. You'll see it written in a number of different ways. Sometimes they put the C's together and the O's together and the H's together, so it'll look like C2H3O2H or CH3COOH, they all mean acetic acid. And when you remove a proton, an H plus, from that, you're removing the H and the plus, you end up with acetate, shown in blue here, CH3CO2, with a minus sign on it. And you can't just buy acetate by itself. It's an ion. It has a charge that you can only buy neutral compounds. So we would typically buy a salt of that base, of that anion. In this case, I've written sodium acetate. You could buy potassium acetate or ammonium acetate. There are a number of different salts that you can get as long as you produce acetate in the process when it's dissolved in water. And when you mix these two together in the right ratios, you can get a very specific pH that is stable even if you add a little bit of strong acid or strong base to it. You can also start with a weak base and add its conjugate weak acid, which will frequently be positively charged, so you can't buy the weak acid by itself. You have to buy the salt of it. So in this case, we have ammonia in blue, NH3, and ammonium ion, NH4 plus, because it's an extra proton has been added to it. That's what makes it a conjugate acid. And we would buy, say, ammonium chloride or ammonium bromide or ammonium hydroxide, something that has ammonium in it, so that when you mix ammonia and ammonium together, you get a buffer. Buffers can't be made with just a strong acid and a strong base mixed together. You can't make buffers that way. And the reason is you need to have a little bit of each, a little bit of the acid component and a little bit of the base component. And strong acids, when you put them in water, they completely form the conjugate base, 100%. And that's not going to work. And strong bases, when you put them in water, completely form their conjugate acid, 100%. So they're useless as a buffer. However, there are tricks where you can use small amounts of these added to weak acids or weak bases, and I'll discuss that in the next slide, where you can use those to produce weak acid and weak base combinations to make your buffers. So here's a generic buffer, okay? It's not, HA is not a real acid. It's just a generic acid. It's something that will release an H+. And I'm showing you the example. When you put it in water, it's going to react with water. It will donate a proton to the water. Remember, protons aren't just freely released. They have to bind to something. And if water is the only base available, water is a very, very weak base. It's also a very, very weak acid. And water is your backup base when you put acid in water. So water will be your base, which will pick up that proton from the HA acid, and you'll end up forming hydronium ion, H3O+, and the conjugate base of HA, which is A-, and A- may be very different it's going to be different depending on which acid you start with. So if you add a certain amount of strong acid to the system, well, that extra acid will react with the base component of your buffer. Remember, there are only two components of the buffer itself. The HA, which is the conjugate acid, the weak acid, and A-, the conjugate base, the weak base in your mixture. So if you add strong acid, it will react with the base, which is A- in this case. And in this particular case, doing that will just make this reaction move backwards. You can see the way it's originally written here in black. It's a reversible reaction. You'll have a little bit of HA, you'll have a little bit of A-, and the A- will act as a sponge to react with any strong acid you add. And when it reacts, it will produce the compounds on the left side here. Likewise, if you add hydroxide, so now that's a strong base to the system, hydroxide is a strong base, it will react with the acid component of your buffer, the weak acid, which is generically HA in this formula. And it will drive the reaction sort of to the right. It's not exactly the way it's written here. We'll go over specific examples that show you exactly what happens. But the OH- will remove a proton from the HA buffer acid and produce more of the conjugate base in the process. So buffers, you're going to start with a weak acid or a weak base, and you're going to add its conjugate, or you're going to make its conjugate. So there are two basic ways to do this. You can either just buy a weak acid and buy the salt of the weak base, or buy a weak base and buy the salt of the conjugate acid. But in other words, you can just buy the two components, mix them together in water directly. The other way is a little trick that chemists and biochemists use all the time. If they only have one of these two weak components, the weak acid or the weak base, you can turn some of the weak acid into a weak base by adding strong base to it, but only a little bit of strong base, and I'll show you that in a second. Or you can start with weak base and turn a little bit of it into weak acid by adding a little bit of strong acid. So here are the standard ways where you're making it directly. We can just mix the conjugate acid and the conjugate base. So in this case, we're starting with acids, weak acids. I've got acetic acid in red on the top and carbonic acid shown on the bottom. That's H2CO3. Carbonic acid is the acid that makes your soda acidic. And acetic acid is the acid that is the component of vinegar. In fact, the word acetic acid, the acetic comes from an ancient Latin word that literally means vinegar. So we start with acetic acid and we add in this case, potassium acetate. Could have added sodium acetate, something that will produce acetate. We mix them together and we get a buffer. We can start with carbonic acid, which you'll notice has 2 protons, but we're only going to be talking about removing the first one. And so if you remove one, you end up with bicarbonate. That's the blue HCO3 minus. Bicarbonate is also called hydrogen carbonate. It has a single negative charge on it. And if you mix, say, carbonic acid with sodium bicarbonate, which you may or may not know, sodium bicarbonate is the chemical name for baking soda. And if you mix these two together, you get a different buffer. You get the carbonate buffer. In the first case, we get the acetate buffer because the buffers are named after the base component. But we can also make these in a tricky way, which is actually very useful in the lab if you only have one component, because everybody in every lab has strong acids and strong bases in the lab. So if we start with just the acetic acid, our weak acid, and we add a little bit of strong base like KOH, we can turn some of our acetic acid into acetate, in this case potassium acetate, because we started with potassium hydroxide. Likewise, we could start with carbonic acid and add a little bit of sodium hydroxide to turn some of the carbonic acid in red into bicarbonate in blue, and therefore you'll have a mix of both the weak acid and the weak base. And this is a perfectly reasonable way to make your buffer. We could also start with the bases instead and do the same type of process. Here's ammonia, NH3 in blue, that's the weak base. We can buy the conjugate acid, ammonium chloride, mix them together and make ammonia buffer. We could do that with a different weak base, like pyridine. Now, you're not responsible for the structure of pyridine or the formula. I'm just giving you an example of another weak base. This is a compound with a ring with nitrogen in the middle of a 6-membered ring, 5 carbons and 1 nitrogen, and the nitrogen acts as a base. The basic part of this base molecule. And when it picks up a proton, you get what's in red here, and we call that pyridinium. And since it's chloride, there's a chloride salt, we call this pyridinium chloride. Again, you're not responsible for the names here. Pyridine and pyridinium chloride, you can mix those together and make another buffer. Or we can do the cheating way, which is just to start with ammonia or pyridine, and add a little bit of a strong acid like HCl, hydrochloric acid, and turn a little bit of our ammonia into ammonium, in this case ammonium chloride, since we're starting with hydrochloric acid, or we can turn a little bit of our pyridine into pyridinium ion. So just by adding small amounts of strong acid to a weak base, we can turn some of that weak base into its conjugate acid, and therefore we'll have a mix of both weak base and weak acid in the end, and that is allowing us to produce our buffer. So let's get back to our generic formula for a buffer. We have the conjugate acid, HA, and the conjugate base, A minus. And this could either be starting with a conjugate with a weak acid or starting with a weak base. Just remember the one with the proton, the extra proton is the conjugate acid. The one with the missing proton is the conjugate base. And let's look at actual examples. Here's a real buffer. This is a buffer where the acid component is nitrous acid. Don't confuse this with nitric acid, which is HNO3. That's a very strong acid, and there's no equilibrium there. We don't have the arrows going back and forth. Nitric acid, all of it goes to the right side of the equation. But nitrous acid is a weak acid. You get some nitrite formed. That's the blue ion here, NO2 minus. That's our conjugate base. And so if we mix nitrous acid with nitrite ion, we make a nitrite base. And likewise, we could do this with ammonia. We can start with ammonia and ammonium, mix them together, and we get another buffer. So I just want you to see how HA, the conjugate acid, could be any number of formulas that have a proton that can be released, and A minus, the conjugate base, is just the same formula with an H removed and with a positive charge removed. And if you get confused about charges, just remember, removing a positive is the same thing as adding a negative charge. So you can see in the first example, in the first real buffer, number 1, the nitrous acid, it's a neutral acid, and when you lose a proton, the conjugate base is negatively charged, whereas in the second case, the acid is ammonium with a positive charge. When you lose the proton, it becomes neutral, ammonia, NH3. So just remember, the charge of the acid will always be one more positive than the charge of the base, and it will have an extra proton. So buffers act to scavenge either added acid or added base. So if we add an acid, a strong acid that's going to produce protons that will really produce hydronium ions, H3O+, they'll be scavenged by the base component of our buffer. And if we add strong base that will produce hydroxide in our water, that will be scavenged by the acid component of our buffer. So what does that look like when we actually do reactions? Well, what it means, first of all, is that if these ions, the hydronium ion and the hydroxide ion, are scavenged, they're basically removed before they change the concentration of hydronium ion in the water, then the pH won't change, because remember, pH is dependent on how much hydronium ion is in the water and how much hydroxide ion is in the water. So if you remove those, if you use the buffer to remove those, then the pH won't change by much, only a tiny bit. And there are hundreds of known commercial buffers you could actually buy. There are probably thousands of known buffers, and if you really tried, I bet there are actually hundreds of thousands of actual buffers out there. And each buffer works only for a narrow range of pHs. So you can't use a buffer for all pHs. Each buffer is good for a 2 pH range. For instance, acetate buffer, which is acetate, that's the base, and acetic acid, that's your weak acid, that works best between pH 3.8 and 5.8. Notice the 2-point range. Ammonia buffer, made from ammonia and ammonium, that works best between 8.3 and pH 10.3. So again, a 2-point range. Bicarbonate buffer, which is bicarbonate and its conjugate acid, carbonic acid, works best between 2.8 and pH 4.8. And phosphate buffers are derived from phosphoric acid. And if you remember earlier in the unit, phosphoric acid has a total of 3 protons it can release. And these 3 protons come off one at a time at different pHs. The second proton is the one that we're going to focus on. And it turns out when you lose the second proton, that happens between pH 5.8 and pH 7.8. So buffers using those versions of phosphate, they work best between these 2 pHs. But there are 2 other pH ranges that certain phosphate buffers will work at. One that's much lower than this and one that's much higher than this. So again, let's go back to the generic buffers here. And let's look at a real example. This one is made from propionic acid. Propionic acid is just like acetic acid, but one extra CH2 group in the middle. It doesn't change the chemistry that happens, it just makes it a longer molecule. So if we start with propionic acid in red, its conjugate base is propionate, and so we call the buffer by the base, propionate buffer. And you see the propionic acid reacts with water, produces the propionate ion in blue, and hydronium ion. So that's our buffer equilibrium. So what happens when we challenge it? Well, let's talk about first how it relates to our generic form, because this is something different that you haven't seen before. So the A is everything but the H in the propionic acid in red. And we'll just throw in the H here so you can see it. And on the A is whatever's left in the conjugate base on the right in blue. So if we add strong acid, H+, which becomes H3O+, of course, that will be scavenged by the base component of the buffer, that is, in this case, the propionate in blue, and it will react with it. Scavenging it means it will react with that component. So we have this reaction where the base component will react with added acid. and that will produce more of the acid component. So we're using the acid we add to just change our buffer ratio so that we have a little more of the acid component in the end, in red, the propionic acid, that was formed from the base component in blue. And in the process, look what happens to the H3O plus. It's turned into neutral water, which has no effect on the pH. And that's how the buffer works to scavenge added acid. It uses its base component. And if we look at the other case, if we add strong base like hydroxide, that's going to be scavenged by the acid component, which we write as HA generically, sometimes AH generically, but we have a real one. It's propionic acid, so it will react with the acid component to form more of the base component. And in this case, it will form the base component. It will remove a proton from the acid component in red and form the base component in blue. And look what happens to the hydroxide. Well, the hydroxide is also turned into simple water in the process. It's gaining an H and a plus. It becomes H2O. So again, no change in pH if you have these two buffer components, the weak acid, which scavenges base, and the weak base, which scavenges acid. Now, I want you to notice in the first case, when we're scavenging acid, this reaction is exactly the opposite of our original buffer equilibrium shown at the top with the green heading propionate buffer. If you look at that heading, that reaction just written backwards with a single arrow is what happens when you add acid. You're literally just driving the reaction backwards. But when you add hydroxide in the bottom case here, it's not exactly the opposite reaction. It's not exactly the same. You'll notice you're not starting with water and ending with hydronium. You're starting with hydroxide and ending with water. But in both of these cases, whatever ion you add, the strong acid ion or the strong base ion, they're converted by one of the two buffer components into simple water. So no change in pH, or a very small change. So let's talk about buffers in living systems. Now, buffers are important in every cell of your body, as well as a lot of the compartments of your body, your small intestines, your large intestines, your stomach. They all have their own pH, and those are controlled by different processes in your body. And your blood is no different. Your blood is controlled by 3 different buffering systems. And so these are the hemoglobin buffering system. At the top, we abbreviate hemoglobin HB. Look in the title here. PI is the abbreviation for phosphate, so there's a phosphate buffering system. And bicarbonate buffer, which we talked about a little bit, but we're going to talk about more in detail here. So these 3 buffers combine to control the pH of our blood very, very carefully and to a very narrow range. Our blood in humans, the pH of a healthy person is always between 7.35 and 7.45. That's a very narrow range, only 1/10 of a pH unit. So 7.40 plus or minus 0.05. And that is done by these 3 buffer systems. So oxygen is transported by hemoglobin. That's not the buffer. The hemoglobin itself is the buffer. The hemoglobin has many weak acid and weak base groups on the protein itself. Protein has many different amino acids that have groups on them, side chains on them, that act as weak acids and weak bases. So hemoglobin is this multi-buffer that helps buffer the pH of the blood. We also have phosphate buffers, and in this case, we're looking at the second proton that is removed. So we start with H2PO4 with a single negative charge. That's the one with the extra proton. And we remove one proton to get its conjugate base. That's the second phosphate form shown here, HPO4 with 2 negative charges. The first one is called dihydrogen phosphate, and the second one's called monohydrogen phosphate. And if you have a mix of these two, you get a pH that's very close to neutral, around the pH of human blood. So this is very useful. And in fact, those of you who are planning to be in medical fields like the nursing students, you'll know that when you give a patient saline just means salt solution, NaCl, sodium chloride, you can't just put water with salt in a person's body most of the time because the blood has to be a certain pH, and you can't just put plain water in there. Sometimes you can, but you frequently want to use water that has a buffer in it. So people will have these IV bottles that will have what's called PBS, phosphate buffered saline. So it's a mixture of these two ions as well as sodium chloride. And there are also potassium ions and sodium ions in there because you need those to counter the charges of the phosphate ions. But this buffer works very well in saline solutions as well as in your blood. And the third system is the bicarbonate buffer system. Well, where does that come from? You need two components, the carbonic acid and the bicarbonate ion. So where does the carbonic acid come from? Well, it turns out that when you eat food, you break down fats, you break down sugars, you break down proteins, you will produce carbon dioxide. And that carbon dioxide, before you exhale it, is swimming through in your blood. Blood has a lot of water in it, so the CO2 will actually react with the water in a reversible reaction to form carbonic acid. This is a combination reaction. It's not the buffer reaction. It is the way you set up the buffer. It's producing one of the components of the buffer, the carbonic acid. And the carbonic acid, being an acid, can release a proton to become bicarbonate. Bicarbonate HCO3 minus. And the mix of carbonic acid and bicarbonate is called a bicarbonate buffer. And in this case, we refer to it as the bicarbonate buffer system in living organisms. And you'll notice I'm writing this in the shorthand that you will find in most other videos where an acid releases a proton, an H plus, and its conjugate base. But the reality is it's a slightly more complicated than that. It doesn't just release the proton freely. The proton is transferred to water to produce hydronium ions. So this is the more proper way to write the bicarbonate buffer system, or any buffer system. You show the acid reacting with water to produce hydronium ion, and the acid becomes its conjugate base, in this case bicarbonate ion. So these are the three buffer systems. The most important one to know for this unit is the bicarbonate buffer system, and with this we'll stop here.